Good evening to everyone joining us from our local time zone, and a warm welcome to those attending from across the globe. I'm thrilled to welcome you all to the 34th edition of the Mentoring Talks series, proudly hosted by the American University of Beirut Transformative Education TRED program. Empowered by students, TRED's initiative stemmed from the belief that knowledge is not a destination, but a journey to be shared. TRED is a dynamic platform branching beyond conventional academia. It encourages experimentation, innovation, and it unleashes creativity. Our aim is to promote students to becoming not only learners, but change makers. Our mentoring talks are not merely traditional lecturers. They are a space where science and experience blend, delving into not only milestones, but the minds that venture to achieve them. They are lanterns shaped by experience, offering novel insights from those who have dared to crave new paths in their fields. As we gather tonight to connect rising minds with thriving scientific discoveries and subdue our collective thirst for knowledge, please join me in welcoming Sir Richard J. Roberts, Nobel Laureate and Chief Scientific Officer of New England Biolabs, a pioneering British biochemist and molecular biologist. His distinguished career has profoundly shaped modern genetics and biotechnology, encompassing both academia and industry over the past 5 decades. This mentoring talk is organized in partnership with the prestigious Angewandte Chemie. I would like to sincerely thank Dr. Frank Maas, Editor-in-Chief of Angewandte Chemie, and Dr. Suzanne Toby, Executive Editor of Angewandte Chemie, for joining us on this panel. After earning his PhD in organic chemistry from the University of Sheffield and conducting postdoctoral research at Harvard University, Sir Richard joined the renowned Cold Spring Harbor Laboratory, where he conducted some of his most groundbreaking work. There, in 1977, he co-discovered the existence of split genes, a breakthrough that redefined our understanding of gene structure and later earned him the 1993 Nobel Prize in Physiology or Medicine. Beyond the laboratory, Sir Richard has leveraged his expertise and reputation to address global challenges, spearheading a coalition of over 150 Nobel laureates, advocating for the use of genetically modified crops to combat hunger and malnutrition in the developing world. Sir Richard's contributions are remarkably broad, ranging from pioneering the use of computational tools in molecular biology to the discovery and characterization of hundreds of DNA restriction enzymes, the molecular scissors that underpin modern genetic research and biotechnology. His achievements have earned him numerous honors and accolades, including election as a Fellow of the Royal Society in 1995 and a knighthood by the late Queen Elizabeth II in 2008. Throughout his career, Sir Richard has exemplified intellectual curiosity, integrity, and perseverance, often stressing the importance of questioning assumptions and learning from failures as keys to success. As we reflect on Sir Richard's legacy, we are reminded that science is not just about discovery, but about purpose, using knowledge to uplift, to question, and to inspire. This should be remembered as we prepare to enter our dialogue with Sir Richard today, opening our minds and hearts together. He once stated, quote, "The education systems in many countries seem to knock the creativity out of children," end of quote. However, Here at AUB, with our thread, we do the contrary. We stand by nurturing intuitiveness, which is why no better individual can help us execute that today. Today, we are given the chance not only to talk to a Nobel laureate, but to an individual whose career stands as a testament to the power of curiosity, integrity, and scientific courage. So, Richard, on behalf of the entire AUB community, we thank you for your worldwide contributions and mentorship. You have elevated the human genome from a mere code, but also as a call to serve humanity. Without further ado, Sir Richard, the floor is yours.
Okay, well, thank you very much. And what I want to do today is to take you fairly quickly through the path that took me to the Nobel Prize, but going along the way, telling you some of the things that I thought were incredibly important in my life and which you may find important in your lives too. I was born in 1943 in Derby, England, shown in the middle on the left-hand side. And when I was 4 years old, I moved to Bath in the southwest of England. Now, when I was in Derby, I lived on the main flight. It was during the Second World War, and I was on the main flight path where the Germans were trying to bomb the Rolls-Royce factory that was making the engines for all of the fighter planes and bombers that were tackling Germany. But in those days, bombing wasn't terribly accurate. And the street I lived on, one side of it was really pretty heavily destroyed, but not the side I lived on. And so on looking back, I see that this was the first piece of luck that I ever had. And I will tell you more about some lucky things going forward. One of the most important things, though, is that luck really, if you get some piece of luck, you must take advantage of it. Don't feel guilty because you had it and no one else did. Take advantage of the luck when it comes your way. Now, I went to a junior school called St. Stephen's School in Bath, and I had to walk uphill in order to get to it every day. It was a small school, but something really important happened there. When I was, I guess, probably about 8 or 9, I used to leave the school in the evening, and the headmaster would come up to me occasionally and would give me a piece of paper with a puzzle on it. And all of these puzzles were mathematical. And as a result of that, I fell in love with mathematics. I thought math was just the best thing going. And if you'd asked me then, I would have told you I'm going to be a professional mathematician. Well, that didn't happen. And it didn't happen because my father bought me a chemistry set when I was, I guess, 10 or 11. And just before then, I'd read a book about how to become a detective. And I was pretty convinced that I was going to be a detective because it seemed such fun. It was sort of like solving mathematical problems, but doing it in an air of criminality. My parents supported my curiosity and love of puzzles. And these two pictures are from the city of Bath in England, just showing you where I was born and where I grew up in Bath. This is the school that I went to where the headmaster got me interested in mathematics. This is just a picture of Bath, England, where I grew up. The street I lived on was very close to this Royal Crescent on the right-hand side. And I was about 11, maybe 11 and a half. My father bought me a chemistry set, and then he built a chemistry lab in the kitchen. You know, we only had 2 rooms in the apartment that we lived in, and the kitchen was quite big, and so he put in a sort of a makeshift kitchen there. And the days at that time were when what was going on in the sort of world for kids was that the chemistry sets you bought were actually very interesting. They had lots of fun chemicals in them, and you could make fireworks and explosives with them. And that really got me very interested in chemistry. And I then went on, and for probably the next 10, 12 years, chemistry was what I loved and what I got involved in. Let's see. So after I finished at the junior school, I went to the City of Bath Boys' School as a grammar school, and it was the sort of high school in the city at that time where kids who were going to pursue an intellectual career were educated from age 11 onwards. And while I was there, I increased my love of mathematics. I learned to play the violin and joined the school orchestra. And I got very interested in caving because it turns out that Bath is very close to a limestone hills, the Mendips, and there were some really interesting and good caves there. And those caves really got me interested in exploration. I love to find new things. I love to go and find a passage that no one had been in before. And I've always had this curiosity and this love of being the first to see something. And now my career is such that I do that every day, and you can't beat it. It's one of the reasons that biology is such fun. We know so little about the basics of biology, and you can always learn new things. Now, for the A-levels, I went to Sheffield University. After A-levels, rather, I went to Sheffield University and majored in chemistry. You had also to do physics, and you had to do mathematics. And I really became very interested in a career in research. And I got my BSc in 1965, and I worked with the professor who was head of organic chemistry at the time, David Ollis. And really, it was great to be in his lab, but not because he was such a great mentor, but because there was a postdoc in his lab called Kazu Kurosawa. And Kazu was probably my first real mentor, maybe during my whole education. He was a Japanese postdoc. By chance, I was put in a lab where he was there, and he taught me more about chemistry than anybody. He taught me how to do experiments, what experiment I should do, how to interpret the results, and most importantly, what to do when an experiment failed. And this is something that I've really valued for my entire life. I just love it when I give a graduate student or a postdoc a project and say, oh, you should do this. And they come back and they say, well, I did what you told me and it didn't work. It failed. And I always view this as being one of the great learning experiences. If you have an experiment, it doesn't work, then you do a post-mortem to find out why the experiment failed. And if you're lucky, it was because the hypothesis upon which your experiment was based is wrong. And you have to revise it, and you have to find something else. And that's always been a very exciting time for me. I love it when things go wrong, and then you go back and find out why and make sure they don't go wrong again. Kazu also taught me how to play the Japanese game of Go, so shown in the bottom left there. And actually, after he left to go back to Japan, we used to play Go by mail, real mail, not email, real mail, for probably about a year and a half, until his English rather deteriorated. And it was difficult, obviously, for him to write the letters that accompanied the next move. But the other thing that happened to me when I was doing my PhD, in fact, I might mention that thanks to Kazu, by the end of the first year, I pretty much had everything I needed for my PhD. I could have written the thesis then. But of course, in England, you have to spend 3 years doing a PhD. So during the last year of my PhD, when Kazu wasn't there anymore, I spent a lot of time in the library, and I came across this book, The Thread of Life by John Kendrew. And this was a book about the origins of molecular biology. And it's one of these things, by the time I finished this book, I knew I didn't want to be a chemist anymore. I wanted to be a molecular biologist. And so when it came time to apply for postdoctoral positions, because I had in mind that I was going to be looking for an academic life. And in order to get a job back in England, you had to go abroad for your postdoc. And so I applied to 6 different laboratories, all of which were run by people who had some interest in molecular biology, but also some interest in chemistry. Now, of those 6 labs that I applied to, only one accepted me, and that was this man, Jack Strominger. And when I applied to him, he was a professor in Wisconsin at the Madison, in University of Wisconsin at Madison. And so I thought I was going to be going to Wisconsin for my postdoc, but just before I was scheduled to leave, he wrote to me and he said, "Oh, could you please delay coming for 2 months because I'm just moving to Harvard. I've just been made a full professor at Harvard." And this apparently, as he told me, this was his career goal. He'd always wanted to be a professor at Harvard. And so when he got to Harvard, he first of all changed fields, and the project I was given to sequence this tRNA glycine was just a sort of a residue of what he'd been working on previously, but he didn't spend a lot of time trying to tell me what to do or anything like that. And I ended up picking up techniques from the postdoc who'd been working on it in Wisconsin. He left after I was there for about 6 months, and he'd been trying to sequence this tRNA using old-fashioned methods. These were methods that had been pioneered by Bob Holly, and they were very good. Holly got the Nobel Prize for it, but they were not the best way to sequence RNA. And I started reading, and I discovered that what was going on in Cambridge, at the MRC lab in Cambridge, was a man called Fred Sanger was actually doing sequencing. The methods that had been developed by Fred Sanger to sequence RNA use P32 labeled tRNA. And I went and I spent 6 weeks in Fred's lab learning how to do his techniques. And Fred shared the Nobel Prize not for RNA sequencing, but for DNA sequencing, together with Wally Gilbert, shown on the left. And of course, Fred had already won 1 Nobel Prize for methods of sequencing proteins. So this was his 2nd Nobel Prize. He should have had a 3rd, because they should have given him one for RNA sequencing, because that was an incredibly important technique also. Anyway, I went and learned how to do that and set that up back at Harvard. And Jack had, fortunately, plenty of money and made one of the lab rooms that he had access to into a sort of a mini MRC sequencing lab. And it turned out I was the first person in the Boston area to be using Fred Sanger's technique in order to sequence RNA. That made me very popular, and I met a lot of interesting people at the time, including Wally Gilbert, who came to learn how to do it, and his technician, Alan Maxam, Mark Potashny, Tom Maniatis, a lot of well-known molecular biologists who were at Harvard at the time, and some who were not at Harvard came to my lab to learn how to do it. And that was very important, and I think there's an important lesson there, that networking and getting to know people who are influential can be incredibly helpful to you. And so when you get the chance to meet people, talk to them, make sure you remember them, they remember you, and you'll never know when in your life this will turn out to be very, very useful to you. Now, I went to Cold Spring Harbor in 1972, but let me tell you a little bit about the lead up to that. When I'd first gone to Harvard and to the USA, I'd always intended to go back to England and to get a job back in England. And I'd been told that there was a very good job available for me in the University of Edinburgh. And in fact, a professor there, Ken Murray, sent me an application form, said he was going to write a letter of recommendation, but he said I was perfect for the job and the job would be great for me. So I sent in the application and I never heard back. In the meantime, I got offered a job at Harvard and then a job at Cold Spring Harbor from Jim Watson. And that job came about because Mark Potashny, who was an assistant professor at Harvard at the time, and one of the people I taught how to do RNA sequencing, he had told Jim about me, and Jim had said he was going to come and talk to me and offer me a job. Well, a couple of weeks went by after Mark had told me this. And then Mark came up to me and he said, well, Jim doesn't know who you are. Perhaps you can go and introduce yourself. So I go along and I knock on his door. And I say, after maybe 10 minutes is a little long. It may only have been 5 minutes. I wasn't in his office for very long, but as soon as I walked in the office, I said, I'm Rich Roberts. And he said, oh, yes, I want you to come and work at Cold Spring Harbor Laboratory. And that was basically the nature of the interview. He just wanted me, and he said, we've got a job for you. And I was just leaving his office, and he said, oh, well, you probably should come down to Cold Spring Harbor and see what it's like down there. But the job is yours, he said. It's not an interview. The job is yours. And so what he wanted me to do was to sequence the DNA of the virus SV40. This was one of the small tumor viruses that was known at the time. And Jim had a cancer research lab down there at Cold Spring Harbor. So that was the job. Well, I took the job and went down there. But when I arrived, I realized there were two other groups, one in Ghent, Belgium, and one over at Yale, that were already trying to sequence SV40 DNA using exactly these same methods. Now, one of my faults, or maybe I don't know whether it's a fault or not, but I don't like people telling me what to do. And I particularly don't like competing with, in this case, two other labs who were doing exactly the same thing that Jim wanted me to do. And so I started to look around, and I remembered a very nice lecture that I heard from Dan Nathans, this man at the top left here, who had won the Nobel Prize in 1970, or later won the Nobel Prize in 1978 for the discovery of restriction enzymes and their applications to problems of molecular genetics. And what had happened is that Dan Nathans had looked at SV40 DNA and shown that this enzyme, endonuclease R, that Ham Smith had discovered, which was the first type 2 restriction enzyme, it cut SV40 into a few pieces, and they were very manageable. And one of the reasons that Fred Sanger had developed RNA sequencing first, before DNA sequencing, was that there were no small molecules to practice on. So plenty of small RNA molecules, but no small DNA molecules. And it seemed to me that this endonuclease, which gave some small fragments that were very small, some of them were only a few hundred base pairs long. And so I thought it should be possible, if there were more of these restriction enzymes, to actually use them to develop methods for sequencing DNA. And so that's what I set out to do. I said, I'm not going to sequence SV40 DNA using the old-fashioned methods and by doing them, copying it into RNA and so on, but I'll develop a brand new method for doing it. And so when I got to Cold Spring Harbor, the first thing I did was to start looking in the various bacterial strains that I could find and using them to see if there were restriction enzymes. And everywhere we looked, we found them. It was really quite incredible. And rather than using them in order to sequence DNA, they became a topic of interest all by themselves because everybody wanted them. And what they wanted them for was to make small gene-sized fragments that could be put into recombinant DNAs, and cloning took place. And then after you'd made the constructs, you could use the restriction enzymes to then look at the constructs that had been made and make sure they'd been made accurately. And so there was a whole new use for them. Now, Jim Watson got very upset about this. He said, you know, I hired you here to sequence SV40 using RNA. I don't want you looking at all these restriction enzymes, and went to great lengths to try to stop me from continuing to work on restriction enzymes. But fortunately, I ran into Norton Zinder, who at the time was a professor at Rockefeller University, and he was a very good friend of Jim Watson. And Norton loved restriction enzymes. And so every time Jim threatened to fire me, I would call Norton, he'd call Jim, and then Jim would say, well, not this time, next time I'm going to fire you. So Norton saved me on many occasions from Jim Watson, but we did end up, Jim and I, not having a particularly good relationship while I was there, because Jim liked people to do things he wanted them to do, and I prefer to do things that I want to do. And I think this is actually rather good in young researchers to look for new things, things that you can do, things that other people are not doing, and to find projects that you can get excited about, rather than just doing things that other people might get excited about. Now, the way in which we sort of looked for the new restriction enzymes at Cold Spring Harbor was every time a postdoc would come in, they'd have a project, they'd be given a new bacterium, and then asked to go and look and see if they could find a restriction enzyme. And very often they found one or two or three, and these were very quick. It was very quick to do this in those days, and you could get a paper out of it. So this is something else that I think is very valuable. When you have new students come in, it's very good to give them what I call bread and butter projects, things that are very straightforward. You know they're going to work, and you know there's going to be a paper that can come out of it. And then they can use the rest of their time to do more interesting things. Now, one postdoc, Richard Gelinas, joined my lab in 1974. I'd met him when I was a postdoc at Harvard and he was a student. And he wrote to me and said, could he come and be a postdoc? And I said, fine. And so he started working with adenovirus 2 messenger RNA. And the question that he asked was, do eukaryotic promoters look like prokaryotic promoters? These sequences that tell the RNA polymerase where to start making RNA is called the promoter. And these had been discovered they'd been discovered in many places, but they'd been sequenced at Harvard in two labs, one in Wally Gilbert's lab, one in Mark Potashny's lab. And we were just curious. They looked very similar to one another. We thought, well, in adenoviruses, which is a eukaryotic viruses, are the promoters the same, or are they different from prokaryotic promoters? So, and we set out to do the following experiment. We knew that there were 4 RNAs, basically, that were being made early after adenovirus transcription, after adenovirus infection, rather, and they were coming in from the ends of the adenovirus, which is a linear virus. And so we thought we'd try and find the 5' ends of these messenger RNAs and then map them onto the sequence of adenovirus 2. Then I had another postdoc who was sequencing the ends of adenovirus. Well, we couldn't find, unfortunately, the ends of the E1 and E2, E3, E4, the early RNAs, because there just wasn't enough RNA there. And it was very difficult to isolate enough to do the experiments that we wanted to do. So we decided to look late in infection. And late in infection, there are a lot more RNAs being made, and they come from all over the genome. And so I came up with a method, a proposed method, that we could find the 5' ends of all the messenger RNAs by taking advantage of the fact that there was a cap structure at the 5' end. And I developed a method, or came up with a method, that would allow us to capture all of these 5' ends. Now, we knew there were about a dozen late mRNAs, and so when we captured the 5' ends, we expected that there'd be about a dozen spots. But in fact, there were not a dozen different sequences, there was only one. It turned out that all of the late adeno RNAs had exactly the same 5' end, which was not what we were expecting. And so, our expectations resulted in a failure, really. And this was a tremendous failure, and we had to do a post-mortem to find out why. And in the process of doing the post-mortem, we found that this one predominant 5' undeconucleotide, so an 11 long sequence at the end of all of the adenovirus late messenger RNAs, we found out where it was, but we couldn't understand how it could get onto the end of all the different mRNAs. And so one possibility was that it was a primer, that there was a separate RNA that was priming the synthesis of all these messages. And that was what I thought was the most likely thing. It was a hypothesis we talked about for a long time, but we did a lot of experiments. We spent probably about a year, not just Richard and I, but some other people in my lab too, looking to see what was going on and was there some explanation for this. And eventually we got to a point where one day I thought of an experiment because we thought if there is a primer, if the RNA has a priming spot, then that primer, while it may hybridize to a bit of the sequence immediately preceding the main body of the message, probably won't be there altogether. And we knew that there was a small RNA made by adenovirus called VA-RNA, and I thought that might be the primer. We knew where it was coded, and so we sat and set up an experiment in which we looked to see whether or not we could find this 5' primer. And if you look at the top right-hand picture here, you will see that what is going on is that if you take double-stranded DNA, melt it, and add RNA to it, you can form what is called an R loop. And this is stable for the simple reason that DNA-RNA hybrids are stronger than DNA-DNA hybrids. Now, because we were looking at messenger RNAs, we knew at the 3' end there was this big poly A tail. And so we expected to see that sticking out from the R loop. But if the 5' end was also coming from a primer that was not coded immediately adjacent to the main gene, then it too would stick out at the end. And so we would expect to see, if we used an appropriate probe, a piece of DNA that could match where the primer was coming from, we would expect to see this picture shown in the middle, that the tail at the 5' end would have the DNA hybridized to it that was coding for that 5' end. Now, I thought of this experiment on a Saturday morning, and Richard and I thought about it, but it was an electron microscopy experiment, and neither he nor I were electron microscopists. But fortunately, two excellent microscopists, Tom Broker and Louise Chow, worked in a lab rather close to ours at Cold Spring Harbor, and we went down and talked to them and say, hey, is it possible that you could do this experiment for us if we make the reagents that you need? And they looked at it and they said, well, it's never quite been done before, but yes, we could do this. This should be okay. And so Richard and I set about making the probe DNA, making the R loops, getting everything set to go. And then on the Tuesday morning, Louise Chow did the experiment and saw exactly what we predicted and looking at that middle screen, with one small exception, and that was that instead of there being a single point of hybridization, there were actually 2 points of hybridization, as shown at the bottom. So the DNA hybridized to part of the 5' end, but then another part of it was hybridizing to the rest of the 5' end, and so there was a loop. So in fact, a typical messenger RNA in adenovirus had 3 separate segments, the main body, and then these 2 loop regions ahead of it. And this was the discovery of RNA splicing. It turned out every molecule that Louise looked at looked like this, so it wasn't just an odd fictional fragment. You know, it was one of the problems with electron microscopy is you only see 1 or 2 molecules out of many hundreds, thousands, even tens of thousands, and you have to be sure that what you're looking at is not some small artifact, but is in fact genuine. But in fact, in our case, we'd already done all the biochemistry to show that everything was the same, and so this was really a discovery. So by the Tuesday afternoon, we were going around telling everybody about this result, and they were all talking about Nobel Prizes. It was really a very exciting time. So we did this experiment, got the results. This was the paper that came out of this. It was a paper in Cell called An Amazing Sequence Arrangement at the 5' ends of adenovirus 2 messenger RNA. Now, I'll tell you an interesting story about this, and that is that while we were submitting the paper, I'd spoken to Ben Lewin, who was the editor of Cell, arranging for this paper and 3 more from Cold Spring Harbor that bore on this same thing. And he was very excited, and it went for review. We got the reviews back. All the reviewers said, this is a great paper, but you have to change the title. It's not appropriate to use the word amazing in the title of a scientific paper. And so when we got these results, I called Ben Lewin and talked about 1 or 2 other things that the reviewers had been, had said, just things, suggestions they'd made to improve this or that or the other. And during the conversation, I said to Ben, when you first heard about this, what did you think? And he said, oh, it was amazing. I said, I rest my case. And so he allowed us to use the word amazing in the title of this paper. And so that's another interesting thing there, to know that sometimes it's worth talking to the editors of papers if they say something that you don't like, especially. And they can often change their mind, and they can be very good to you and allow you to do what you want to do rather than what the referees decided you should do. So this was a very exciting discovery. And I should say that at the same time, I think actually it was maybe a week or 2 after we had made the discovery, Phil Sharp made the same discovery independently up at MIT. And then everybody started when they heard about it, they started looking at their data and realized many of them, they had data already that they'd not been able to explain until they knew about RNA splicing. So this really started a lot of people getting very interested in RNA splicing. Now, as I say, Phil and I won the prize in 1993. And one of the things you have to do when you go to Stockholm is to give a lecture, describing whatever you want to talk about. And most people, when they give those lectures, talk about all the fabulous work that they've done following up on the discovery, which is what Phil did. But I'd actually stopped working on that area many years before and had gotten interested in DNA methylases in bacteria instead. And so I couldn't talk about splicing, but I was lucky. Just a couple of months before I had to go to Stockholm, one of the things that happened was that Jiadong Cheng, who was a postdoc at Cold Spring Harbor at the time, he and I had been working on trying to crystallize DNA methylase. And he got the structure of MHHA1 binding to DNA. And what we discovered was that the base that was going to be methylated was flipped right out of the DNA and into the active site pocket in the enzyme. And so I made that the focus of my talk in Stockholm. I figure if you've just made a big discovery, there's no place better than the Nobel Committee to talk about it. And so that's what I did. And we also noted at the time that there were probably many other areas where enzymes that do chemistry on DNA use base flipping in order to get access to the bases, because otherwise the bases are buried in the helix. And we now know that that is true, that almost every enzyme that does chemistry on DNA uses this base flipping mechanism to get access to the base. I'm going to switch completely now and tell you about something that followed up from the work at Cold Spring Harbor, and that is New England Biolabs, where I now work. New England Biolabs was founded by Don Combe, shown on the left, and in 1974, or late 74, early 75, he changed the name from Biolabs to New England Biolabs, and he started to think about selling the first restriction enzyme that Ham Smith had discovered, but he was going to sell it to a distributor, Miles Laboratory. Now, I had heard about Miles. I called them up and I said, you know, I'd found a lot more restriction enzymes. At the time, we had 30 that we'd discovered. And I tried to convince Jim Watson to set up a company at Cold Spring Harbor to sell these enzymes and use them, you know, as a source of income to support the research at Cold Spring Harbor. But he poo-pooed that idea. He didn't think it was very good. He didn't think it was likely to happen. Kept telling me it was disgusting to be associated with industry, and you know, this was not what academics did. But at the time, I was giving all these enzymes out to scientists around the world. They all came to Cold Spring Harbor. They wanted to see if they'd bring DNA in their pockets to find out if we had an enzyme that could cut it. So I knew there was a business there. So anyway, so I called Don Combe, and he said, oh, come up and talk to me. And at the time, New England Biolabs was really in the basement underneath a hairdressing salon in Beverly. And the company consisted of Don Combe, his wife, and one technician. And they were doing pregnancy testing, and they were making 1 or 2 enzymes that they were selling. And he was also doing some lobster fishing. And this was how he was keeping his family going. But he'd always wanted to set up a company to make money and then to use that money to support research. And that is what he's done at New England Biolabs. I joined him as employee number 4, although just as a consultant. And I sent enzymes to him, told him which ones he should make. And in the first year, we made $200,000, which in 1975 was a lot of money. And it was very good. In 92, I moved up to New England Biolabs as a research director, and I'm now the chief scientific officer. And the company at the time, we put up this building in North Beverly in 1984. And in 2005, we put up another building because we'd outgrown the first one, which is where we are today. So New England Biolabs has been very successful. When I went there, I was the 4th employee. We now have about 750 employees worldwide. So we've grown quite a lot, and we've been very profitable. And we use the profits to support research. We have about 100 people just doing research, nothing but research. They could be an academic lab. They have to publish everything. And we give them a lot of freedom as to what they want to work on. Now, I'll tell you about 1 or 2 other things that have come up since I won the prize. I spent my money on a croquet lawn in front of my house because I love playing croquet. And it was the only way I could ever have afforded it. So that was rather nice. And it's good because it combines the skill of billiards and snooker with the strategy of chess. And I also have a dry sense of humor, I might say. And I featured as Dr. December in the 1997 calendar, the Stud Muffins of Science. And I go down to the Ig Nobel Awards every year when I'm here. They're really a lot of fun. But I love puzzles, I love games. Now, I've also had a few honorary degrees, probably more than I need, but they keep coming. One of the science departments at the school, City of Bath Boys School, the grammar school I went to, was named after me. And a large expansion to the chemistry department of the University of Sheffield was also named after me. They rather liked me there because I was their first student ever to win a Nobel Prize. As you heard previously, I was knighted by Prince Charles, actually, in Queen Elizabeth's 2008 birthday honors list. And I've helped to organize a lot of the Nobel laureates for good causes, including getting some Bulgarian nurses out of jail in Libya. We started this when I discovered that once you win a Nobel Prize, people listen to you. And sometimes they don't just listen, they act on what you suggest. And so the first sort of foray into using the Nobel laureates and organizing them was about open access to the literature, which was a long time ago when we started it. But now you can see that open access to the literature is becoming the norm. And I think this is really important that, you know, we do the work we do, we get paid by usually government grants. So the people are paying us, and then they shouldn't have to pay in order to read the results that we've managed to obtain. And I want to finish by talking about the importance of luck. So I first found out about this playing billiards, well, actually playing snooker, I was the West of England junior snooker champion, and I was playing one day, and I tried to pot a ball, and I missed, but I potted something else instead. And instead of taking advantage of that, I messed up the next shot. I wasn't very careful, and so on. And afterwards, an old man came up to me, must have been 80 years old, came up and he said, "You know, you're a very good snooker player," he said, "but you have to learn that when you have a lucky break, you've got to take advantage of it. You can't just let it go. You don't feel guilty about it. You concentrate twice as hard on the next thing you do." And this is advice that has stayed with me and which I will pass on to you because I think this is really important. Kazu Kurosawa, I told you about, the best mentor I'd ever had. Going to Harvard instead of Wisconsin, that was luck. Looking for eukaryotic promoters in adenovirus. We never really characterized the eukaryotic promoter there other than to get it sequenced, but we discovered splicing because our experiment that we did failed, and we looked to see why. I've also been very lucky taking an early plane. You probably all remember of the attack on the World Trade Towers in New York in 2001. And the first plane to go into the World Trade Tower, I was booked upon that until 2 weeks before it took off. And I was going to a meeting in California, and the organizers moved the meeting forward by 1 day. And so I ended up having to change my reservation and taking the plane 1 day earlier. And so I was actually in California and not on the plane that went into the World Trade Tower. So that's pretty lucky. And then finally, studying bacterial RM systems. The restriction enzymes were great. They led to really, they formed the backbone of the biotech industry back in the 1970s and 1980s. They helped form New England Biolabs, and they got me a lot of papers, a lot of recognition, and I started a database exactly 50 years ago called Rebase, in which we keep track of all of the restriction systems, the enzymes, the methylators, and so on, that are known today. So at this point, I will happily pass on this and say I'm open to any questions that you might have. So thank you very much.
Thank you so much, Sir Richard, for this beautiful and inspiring mentoring talk. For all the attendees, you can write your questions in the Q&A inbox, and I'll be reading the questions, and Sir Richard will answer them.
If I can.
Try.
I'm hoping for some that I don't know the answer to.
So the first question is from Mazen Abdul Samad. He's biology majoring student here at AUB. The question is, what drives you to keep moving forward during difficult moments, and how do you stay connected to your purpose when the path ahead is not really clear?
Well, I think when it's not really clear is actually the most exciting part of research, because it means you have to spend a lot of time thinking of possibilities and things that you might do to go forward. So I rather like that aspect of research when you don't know exactly what you should be doing. Maybe you've done an experiment, it didn't work, you want to know why, and you have to keep thinking of new ways to find out why. And for me, science is my hobby. You know, I don't think I've worked a day in my life. And I recommend all students, find something that you love to do, that you say on a Monday morning, I've got to go to work, it's great. And on a Friday afternoon, you say, what am I going to do until over the weekend until Monday comes around again so I can get back to work? And these are the people who are really happy in life. And if you're happy in your job, the amount of money you make is not terribly important, you know, provided you have enough to keep your family going. And I think you'll find that the family you'll get on better with your family if you're in a job that you really like. They don't want to hear you come home and complain every night, oh, I had to do this all day today, I had to do this all day today. It's nice when you go home and you'll come home happy, and that's the way to deal with the family, I think.
Thank you. This question is from Hassan Abu Dahir, chemistry student at AUB. In your opinion, which qualities are necessary for a good mentor in the field of science?
Well, I think this is something that is really, it's something that's very difficult for a lot of people. What you need when you're doing science, and at least in the early days, is you need someone who can explain to you very clearly what experiments are worth doing, why they should be done, how they should be done, and then can help you to interpret results. And that means someone who will actually spend a lot of time with you. And this is why other graduate students and postdocs often make the very best mentors. The professors, they're too busy writing grants or doing other things, and they don't have enough time, usually, unless they're right at the start of their academic careers, they don't have the time to really teach you exactly what should be done. And it's tough. They like to see the results, but they don't often prove to be very helpful in the actual mechanics of doing the experiments. So for me, that was why Kazu Kurosawa was such a great mentor. I mean, he was just terrific. We'd sit and talk, and he'd explain what the experiment should be done and how to do it, and then you could interpret the results, and he was just great. So,
Thank you. This question is from Lara Mirai. She is an environmental health measuring student. Her question is, you have experienced both academic and field domains. What advice would you give to scientists to be able to excel in both?
Well, I'm not sure that I can advise you how to excel in both, but I can tell you how to enjoy both. And the enjoyment comes really from looking either out in the field or in the lab, seeing what is interesting, and then starting to ask questions. So use the experiences you have to drive your curiosity and to make you start to think about experiments. You know, these days, everything I do is bioinformatics. So I sit in front of a computer screen and I look at output from programs that I help to create, and I see new things, and I want to ask questions, and I want to see, oh, you know, what does this mean? What does this mean? And for me, this is great. Just having an inquiring mind, having curiosity is very important. And as you said in your introduction, when we go to school these days, very often young people, they have the curiosity knocked out of them because the teacher says, oh, you've got to do this, you've got to do that, you've got to do it exactly this way. And we had an interesting conversation with my youngest son, who his math teacher kept giving him low marks, even though he was getting the right answer. And he was giving low marks because he wasn't doing the problems the way the teacher had taught him to do them. Rather, he developed his own way of finding the answers to these questions. And after we had this talk with the teacher agreed that it was okay. And so my son started getting decent marks. So not everybody works in exactly the same way, and imagination, doing things a little differently, this can often lead you into new problems, new findings, new discoveries that can be very exciting.
Thank you. We are going to keep it, as I promised you, only 15 minutes past the hour. So 11 more minutes, if you don't mind.
Okay. Okay.
Thank you so much for your time. And I know that also Dr. Frank and Dr. Suzanne, we're keeping them very late. They had a very busy day today. Doctor, this question is coming from Leticia Nassar. Hopefully she will become doctor soon. Her question is, what advice would you give to young researchers who aspire to make significant contributions to science?
Well, I think the first thing is you have to find what field of science that you're most interested in. And the way in which I always advise people here is to read widely and go to many different lectures. So, you know, I've found times when I've gone to a lecture not in my field, quite away from it, and yet I learned something during that lecture that, you know, I wasn't expecting to learn. And then I kept thinking, oh, I could apply that. You know, the Dan Nason's lecture I told you about restriction enzymes. I went because it was interesting. I didn't think I'd use it until I get to Cold Spring Harbor. And then all of a sudden, the prospect of using what I'd learned in his lecture to start. In my case, I thought I was going to develop methods to sequence DNA, but that didn't work out. But the restriction enzymes themselves became so interesting to me, to everybody else. And, you know, I'm still very interested, very fascinated by that. We learn more about this all the time. But you have to find the field that you really want to work in. But don't be afraid. If you're working in a field and you come across something more interesting, don't be afraid to switch, because you can always switch. And I've done that a number of times in my life, and usually it's paid off.
Thank you. This question is coming from Noor Abu Faris, chemistry student. If you could go back in time and offer advice to your younger self, what would you say?
I would say do exactly what you did. I'm really happy with the way that everything turned out. You know, when I was at school, at high school, I thought I'd be a chemist. I kind of had visions of being an industrial chemist and, you know, going to work for one of the big chemical companies in the UK. And then when I got interested, got a PhD, realized that the academic side of life was really very interesting. And then I read this book about molecular biology and stopped. I tell you, you know, I've had this experience a number of times. I've read a book, and at the end of it, I've decided this is what I want to do with my life. You know, not what I'm doing at the moment. I want to do something new. So it's good. It's good. I wouldn't change anything, because I'm pretty sure if I changed something, I probably wouldn't have won the Nobel Prize.
This question is coming from Taleen Sariyuddin. She is a chemical engineering student. What emerging areas in molecular biology excite you the most, and where do you see the field heading in the next decade?
Well, I'm very keen on the possibilities for using artificial intelligence in order to decipher DNA sequences. And what I'm hoping is that before I die, we will understand how a bacterium works, because they're about the simplest organisms we know about. And in order to do that, we have to understand how all the genes work, what they do, what enzymatic action they perform, and how they interact with one another. And I think we could imagine writing a computer program in order to mimic, say, an E. coli cell, and have something that will work in exactly the same way. And I think AI is going to play a big role there. But I'm also very nervous about AI in that the way it's being used at the moment is often not as good as it should be. And the quality of the output that you get depends totally on the quality of the input that goes in. So if you've got a program that is taking data that is not itself perfect and then trying to reach conclusions, it will make mistakes. And we already see this. You look at Chat GPT, I call it Cheat GPT, but you look at these programs and they hallucinate. They produce stuff coming out because the data that they're fed on is not always as good as it could be. And, you know, just saying, oh, we'll use the scientific literature for this purpose is not good enough. There is a lot of rubbish in the scientific literature. We need to pull out those papers that are not that good and use the ones where we know that they're really good. And then I think AI will be able to work much better, but it should also tell us how it reaches the conclusions that it reaches. So, you know, as an experimentalist, when I write a paper, I have a methods section. I say, this is exactly what I did. I think AI programs that are going to, you know, generate output should also tell you how they reach that output. And the computer scientists tend not to want to do that, but I think they should.
Thank you. You actually also answered the question by Christy Ad, who is asking about the use of AI in the future of genetics. Dr. Mas and Dr. Toby, you have questions or you want to comment about pulling old papers that are not of good quality? I hope not in Angewandte, though.
That's what we're hoping for. Yeah. As an editor, I found your story fascinating with your conversation with the editor of Cell about the word amazing in the
Yeah.
title. I also had such conversations from the other side, yeah, with professors. I do have a question in a different direction. So an important message that stuck with me is your advice to do what you want to do and not to
Mhm.
listen to others. So you remember a situation where doing what you wanted to do went wrong and
Mhm.
did not have a good outcome, and you then afterwards said, "Oh, I should have listened to this guy." Um, do you remember such a situation?
Not really. No. I mean, when things go wrong, you know, when you do what you think is a good thing to do and it goes wrong, I like to do a post-mortem and find out why it went wrong, because that's when you learn. I'm always struck by the tale of Bill Gates. You know, the first company he tried to form failed, but
Yes.
Microsoft has been anything but a failure. So this is when you learn, is when you fail. I don't think you learn so much very often by listening to other people. So I like to do things myself. I think when you're very young and you don't have many facts, then you know teaching can be useful. But after a point, it gets to a point where I think you're better off really doing it yourself. And if you fail, then you know, work out why.
Thank you. A couple more questions, if you don't mind.
Okay.
And I'm trying to get questions from different majors. I know a lot of students from my team have put work into preparing questions, and they have researched you. And I have a beautiful team who also prepared a draft of the opening remarks. So they did an amazing job. This question
Did they
is
use
from Leah.
AI for that purpose?
No, 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 I hope not. Well, I could detect that. This question is coming from Leah Slim. She's an environmental health student. How were you able to maintain that spark of curiosity throughout your life?
I think it must be something innate because I like to ask questions. I like to understand how things go on. And I'm always curious about things. You know, I may go out bird watching and I'm curious to watch the birds and see how they interact and what goes on. Or I was in Madagascar with my daughter last year and we were looking at the lemurs there. And it was absolutely fascinating to realize what they are doing that in many ways reflects what humans do. And you start to wonder why. Why is this all possible? So I've always been a curious person, but I guess it was because it was not knocked out of me when I was at school. I think most people are curious, and we tend in the schools to knock it out of them, to tell the kids, no, 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 you've got to do this, you've got to do this, you've got to do this. And if they say why, then the teacher gets annoyed and tells them, you know, stop, just listen to what I say. The question why is a good one. It's a good one.
Two more questions, I promise.
Okay. Time is not a problem. Okay.
Thank you. This question is coming from Jean-Marc Rebez. He's a biology student. How do you cope with criticism and skepticism in your career, especially while working toward your groundbreaking discovery?
Well, I think I tend to ignore it is the answer because, you know, when we first discovered this oligonucleotide, the same one at the end of adenovirus, people kept telling us it was an artifact. I mean, you know, they didn't believe it was a genuine result. And so we kept doing experiments to show that it was genuine. And, you know, I'm not put off by that. I tend to focus on a particular problem and do everything I can to understand it. And, you know, there are many people who like to be critical without any really good reason to be critical. It's just that it falls, you know, you tell them about an experiment you did, gave a result which falls outside of their comfort zone. They tell you it's a mistake, an artifact, and so on. And I think people need to be a little more open-minded, especially as scientists.
Thank you. This question is by Colleen Qubaisi. You have been a vocal proponent of GMOs to alleviate malnutrition.
Yeah.
What are the biggest misconceptions about GMOs, and how can science communication address them?
Well, you know, what happened was that when GMOs were first discovered, we didn't know whether interfering with the genes using the methods that were originally developed were going to lead to problems, because we already know traditional breeding of plants, say, can lead to problems. So now that we have CRISPR and we have very precise methods of altering plant genomes, we can do the tests very easily to see if there are problems. We know exactly what we've done. We've taken one gene and put it in. And so now there's a very different view. To start off with, Greenpeace, who were the principal anti-GMO group, kept saying, oh, you know, we don't know whether this is dangerous. We need to do the science and find out. And they started developing films, marketing materials of one sort and another, deliberately to scare people. And it became the best fundraising move that they ever made. They tripled their budget, their annual budget, just by being anti-GMO. And I think it's very unfortunate, because now, if they were smart, they would say, well, you know, we asked that the science be done to test these. It's been done. We now know they're safe. We've got more than 30 years, and there's not been a single problem, a single accident. And the developing world really needs GMOs. Their crops have never been improved in the way that the crops in the West have been improved, because the agricultural companies don't want to spend the money to do it. They can't make money selling seeds and selling better crops in the developing world. But with GMO technology, you can achieve better crops, better yields, greater nutrition very easily. And you don't need to get the big agricultural companies involved. Scientists in these developing countries can easily improve crops locally. And so I think, you know, it's really appalling that Europe listens to Greenpeace and the anti-GMO people, and it's because they've been so successful in scaring people, you know, and there's no reason to be scared. I'm much more scared about traditional breeding, where you move hundreds of genes from one plant to another. We don't know what most of those genes are. We don't know what they do. We're just looking for one trait that comes out that is better. With GMO, you've got one gene, you move it into the plant and you see what happens. And it's precise. It's a precision method. And it is just so much better than traditional breeding, and it's much faster too. It doesn't take very long to develop a new crop.
And the last question, I think that will be a good way to conclude the session. If you could pass down not a finding, not a publication, nor a discovery, but just one idea for future scientists, what would it be? And how relevant do you believe it will be to them? This is coming from Marie-Josée Daher.
Well, I think, you know, sort of looking forward, I think if we can actually discover how a bacterium works or how a living organism works, and I think bacteria are the best bet, once we get a clear view of how they work, how the genes interact, what goes on, that is knowledge that can be applied to any biological problem. And I think it's going to be very valuable if we know exactly how a biological organism works, because there will be lessons there. You know, already, you know, with gene editing, it is possible to solve diseases in humans. But to understand how a human works is incredibly complicated. It's going to take maybe centuries before we can say we understand how a human works. But a bacterium, I think we will be able to work out exactly how it works in the not too distant future, I say, before I die. And we're going to have so much insight into biology as a result of that, that what we learn will be applicable in plants, in animals, in humans. It's going to be exciting, very exciting.
Well, once again, I would like to thank you, Sir Richard, for honoring us with your time for this beautiful, inspiring mentoring talk. And I hope I will have the chance to host you in person here in our beautiful campus in the future.
Okay.
And I'll be extending you an invitation regarding this for a symposium we are organizing next year to celebrate the 160th anniversary of our beloved AUB. I would like also to thank Dr. Frank Maas and Dr. Suzanne Toby and Angewandte Chemie for their support and for their encouragement. And I want to thank all the attendees until our next mentoring talk on January 27, 2026, which will be our 35th mentoring talk, and it's going to be by Nobel Laureate Martin Shelfie. Until then, thank you all, and thank you, Sir Richard, again.
Okay. You're very welcome. Very, very welcome.
Thank you.
And good luck to all the students. Good luck.
Thank you. You have a great night. Thank you, everybody. Bye-bye.
Thank you, Bilal. Thank you, Sir Richard.